Faculty of Medicine of McGill University has always been one of the most outstanding faculties of medicine in the world.
The last decade has seen huge transformation in cancer research. We've moved to research where we now start with the patient.
In virtually every major aspect of cell and molecular biology.
Really the cutting edge and groundbreaking research equipment and outstanding scientists to fight this disease.
When you have the resources, you have so many more opportunities to attract good people and to do the good science.
There are a very large number of graduate students who are in fact our future in science and particularly in cancer research.
The Cancer Center in this sense is really something very unique.
We have always gone after young people to bring in. They bring ideas together and bring them back to us. Let's try something crazy. Let's give it a shot.
If you have the right people and they're stimulated and they have the passion, you got to support them.
Welcome all to this second in a series of public lectures by the McGill Goodman Cancer Research Center. I'm Joe Schwartz and I'm happy to be your host here today. I direct McGill's Office for Science and Society with a mandate of demystifying science for the public and to separate sense from nonsense. Our subject today is lung cancer, and it's a frightening concept, because this disease is diagnosed a lot in Canada, and the outcome generally is quite poor. But it certainly is not a hopeless situation, because there's a lot of work that is being done, much of it here at McGill at the Goodman Cancer Research Center. Today, we are taping in front of this huge non-crowd because of COVID-19. COVID-19 has affected every aspect of our life, including research, but the research does go on. And we are making some fascinating discoveries. We're going to hear about that today from Dr. Logan Walsh and Dr. Jonathan Spicer, two of the key players in lung cancer research. We hope that you will talk about what you hear today with your friends and your relatives. Tell them to start looking into the research that is being done here because it is very important, and to support it in every possible way because we are now seeing some light at the end of this dark tunnel. At this point, we'd like to welcome our sponsors, and we're very happy to hear from our co-chairs, Jonathan Durocher from Banque Nationale Financière and Paul Sicilian from Bombardier.
Hello and welcome. My name is Paul Sicilian, and I am the Executive Vice President of Operations and Operational Excellence at Bombardier.
And my name is Jonathan Durocher, President of National Bank Financial Wealth Management.
Today's program is brought to you by CDL and ELNA Labs. The Goodman Cancer Research Center is a world-renowned research center based right here in Montreal that focuses its research on hard-to-treat cancers. The center that actually collaborates with the best minds contributes to achieving the goal of improving the outcome of cancer patients and their families. And that is the work that has the potential to impact millions of families. So we're all here watching, and we're very proud to be part of it.
In the current environment, it's important to buy local, bank local, and encourage local industries and research. The GCRC's local expertise falls right in that category, and we are both proud to be associated with their groundbreaking research.
So, without any further ado, please allow me to introduce Mr. Laurent Amram, President of CDL and ELNA Labs, who is going to be the sponsor for tonight's very important public lecture series. Enjoy.
It's our great honor to sponsor this evening's public lecture series. At CDL Laboratories and ELNA Medical, we care about our community by providing quick access to high-quality services by leveraging advanced technologies. Every aspect of our service is designed to achieve better health outcomes for Canadians. The important life-saving work being done at the GCRC and supporting this cause aligns with our values. We are very proud to help advance research through our partnership. Now back to you, Dr. Joe.
Thank you, gentlemen, very much for your ongoing support. Now we get to the highlight of the evening, introduction of our two speakers. Dr. Logan Walsh is Assistant Professor at the Goodman Cancer Research Center and Department of Human Genetics here at McGill. He was recently appointed as the Rosalind Goodman Chair in Lung Cancer Research. Dr. Logan's lab uses both experimental and computational approaches to understand the genetic basis of cancer. The Walsh Lab is focused on fostering close collaborations between physicians and scientists to address clinically relevant challenges. Dr. Jonathan Spicer completed his training in cardiothoracic surgery at the world's largest cancer center, and that's at the University of Texas, the famous MD Anderson Cancer Center. It's in Houston. His clinical interests focus on minimally invasive approaches to lung cancer, as well as complex resections for advanced thoracic malignancies. He has paired his clinical focus with a comprehensive research program in cancer metastasis. Welcome, Drs. Walsh and Spicer.
Thanks, Dr. Joe. Hey, John.
Hey, Logan. Dr. Joe, nice to see you.
Good evening, everyone. It's nice to be with you. I wanted to start off today by trying to put the lung cancer problem into perspective. Right now, we're in the middle of a once-in-a-century global pandemic. It's ravaged our global economy, and it's really crippled our way of life. For the past year, most of us haven't even been able to get together with our friends or even our family members. Now, what if I told you that last year, more people died of lung cancer than died of COVID-19? And not only that, that level of death from lung cancer happened the year before COVID, and the year before, and the year before that. It's been happening for decades. Think about that. Now let me try another approach. More people will die this year of lung cancer than of breast cancer, colorectal cancer, and pancreatic cancer combined. Or another way to think about it, more people will die of lung cancer this year than all the people that die of liver, thyroid, brain, bladder, stomach, kidney, leukemia, lymphoma, melanoma, cervical cancer combined. Does that put it into perspective? Cure lung cancer and you will save nearly 2 million lives every single year. That's on par with the number of deaths from COVID-19 worldwide. COVID every single year. I hope that gives you an appreciation, and solving lung cancer will cure basically 1/4 of all the cancer burden in our country.
Unfortunately, funding for lung cancer is way too low. And that's historically because we haven't had survivors from the disease. No survivors, no advocacy, no funding. It wasn't until a year ago that we had a ribbon for patients afflicted with lung cancer. But things are changing. There's good news. The White Ribbon Project is building steam, and it's bringing awareness about the importance of this terrible malignancy all around the world. Patients are surviving, and we're so fortunate to be part of a team that's passionately focused on altering the course of this disease.
So what are we doing to help with this global crisis of lung cancer? Well, I think there really are 3 modern pillars of cancer treatment. And you really need all 3 of these to give patients the best chance to extend their life or to survive. The first is prevention. That is education, which I hope we're going to be doing a little bit today, but also lifestyle interventions such as smoking cessation, and also diet, exercise, some of the topics that were covered by the last GCRC public forum on obesity, metabolism, and cancer.
The second is early detection, as well as screening programs for high-risk individuals.
That's right, John, and if you were at the last public forum for lung cancer a couple of years ago, what you would have seen was a real-life example of how these screening programs can save lives. It's actually one of your patients, John, that entered into a screening program and detected their cancer as a stage 1. John operated, and that patient is alive and thriving now. I believe it's probably 5 or 6 years later.
And I'm proud to announce that the Canadian Partnership Against Cancer has teamed up with the Quebec Ministry of Health to deploy a lung cancer screening program across our province for Quebecers who are at risk.
That's right, John. And of course, the third pillar is treatment. And this is completely dependent upon research, which allow us to generate new therapeutic options for patients. And then, of course, clinical trials, which bring those new therapies from the lab to the patients that need them.
We're lucky here in Quebec. We have really a great culture and a culture of engagement with our patients and teams that explain to them why what we're doing is helpful.
So maybe we could use the clinical trial as an example of what happens from when a patient comes to the clinic to all the way through the research stuff that we're doing behind the scenes to ultimately, hopefully, double survivorship.
So that patient comes through my clinic. He or she may come through one of my colleagues' clinics, and we have to identify that patient is potentially eligible. This means getting all the diagnostics and staging tests done in a timely fashion and having a picture of where this person's cancer is at to figure out whether they could fit the eligibility for this trial.
We are one of the very few centers in Canada that tests all of their patients for 52 genes. These are 52 cancer predisposing genes that could make them eligible to go on to a targeted therapy. So that's the first part of what's going on here. If they're not, they can become eligible to get onto this clinical trial.
That's right. So that means we're talking with our pathologists, we're talking with our radiologists to get the biopsies. We need enough biopsy to make the diagnosis, but also to be able to do the research. Because the whole point of collecting tissues from patients on trials is that we can see how the cancer actually evolves under treatment over time.
So we've identified the patient, they've come through the clinic, we've done all the paperwork, we've enrolled them on their clinical trial. Now we have a date for them to come in for their treatment. We then, when we identify those dates, we will put in a request to have a blood draw, or if it's the time of surgery, we'll put in a request to have our research team come in at the time of surgery, and they will take a little portion of that tumor back to the lab. We take those portions of tumor or blood cells, and we will study them. One of the things that we do is we actually take a little section of tumor and we can implant it into a mouse, and we can grow it inside of a live animal. It's like a mini patient avatar, and those tumors will grow. And then what we can do is we can take that tumor out, we can chop it up into little bits, and we can put it into a whole cohort of animals to study. And we can treat those animals with various drug combinations to see what drug combinations may be available that work against that specific patient's tumor in a live animal. Tumors will often shed some of their DNA as they're growing, and you can detect that in the blood. And paired with this trial, we are testing over time, just through non-invasive blood draws, whether or not any of that circulating tumor DNA exists in these patients in the months, weeks, and years after the trial. And I think that's an amazing concept whereby you're not waiting for that patient to have some sort of sickness that's identifiable through cough and the tumor has grown quite a bit or spread to another organ that's causing them to have symptoms. What you're doing is on a routine blood check at their clinic on a 6-month or yearly basis, you're able to monitor whether or not they have recurrence, and if they do have some of this circulating tumor DNA detected in their blood, they're probably at early stage of recurrence, and you're able to find out where it is, and you're able to deal with it. And again, the earlier you catch it, the better the patients do. Now, if you multiply that by 50 patients on the trial, now we start to get a much better picture of those that respond to therapy, those that don't, and now we have all the necessary material to study why these patients are responding and why these ones aren't. And that's ultimately the goal here, to try and understand why patients are responding so that we can either administer additional therapies to help the ones that aren't responding respond, and/or we can develop these biomarkers to triage patients that are most likely to respond to these therapies onto the specific therapy regimen for them.
For those of you who were back at the gala in 2018, you got to meet one of my favorite people, John Swade. He's a patient who agreed in 2017, right around this time of year, to participate as one of the first people in the world to enroll in a treatment of immunotherapy before surgery. He put his faith in us. I was only in my second year of practice. It was the first patient I'd ever put on a clinical trial. And he received immunotherapy, went on to get a relatively complicated operation, and is now 4 years later surviving his cancer, still going to work. He's in his 80s. I get to talk to him every few months, tell him his CT scans are clean and doing well. And it's the most rewarding thing in the world to know that these efforts that we've put together and what they'll bring to people now and what will be brought to people in the years to come. It's just amazing, and we're so proud of it. So we wanted to share that with you, let you know that he's alive and doing well.
In December of 2016, I was losing weight, and I went to see my GP in December, and my results came in January that I had a tumor on my lungs. So we went to see Dr. Spicer, and Dr. Spicer told me there's a new treatment called immunotherapy, and I started my treatments, and I continued the treatments, and I continued going to my office and doing work. I never felt anything, weakness or pain. I didn't lose my hair. I had good appetite. Everything was normal. Once we finished the treatment, we did the operation, and it seemed to be successful. And he said, well, further, if I was you, I'd do a little more treatment just to make sure. I was always feeling fantastic, actually. And people couldn't believe that I was going through all these treatments and nothing affected me. I was the first one to do this trial, this chemotherapy trial, first one in the world. I just couldn't believe that I was doing this experiment. And I'd hope if it worked, and which it did, that it would help other people feel a little more comfortable being diagnosed with lung cancer, especially at stage 4. Say that like a miracle. Somebody saved my life, and they're probably part of it.
The last thing I really wanted to mention is that at the Goodman Cancer Research Center, we have all of the latest technology. And the reason why we're able to have all of this technology is through our generous donor population. Things like the GALA. We have imaging, metabolomics, genomics, you name it. We have everything available to do the best biomedical research. So we are on par with the best institutions in the world for technology, and that's why today, the Goodman Cancer Research Center is a leader in cancer research.
We've been able to build a team that unifies this research program throughout our clinics and our labs. And this is what's going to make the biggest difference. I'll just say a couple words about why it's important to have a network. I mean, because it seems we can do it all ourselves here, and we do have an amazing team. But what happens after this trial is finished? Well, say we're extremely successful and we show great benefits to our patient. Those medications can't come to patients as a part of standard of care until we've delivered these drugs through phase 3 trials that are deployed internationally. So we need national partnerships with Marathon of Hope, the Canadian Cancer Clinical Trials Group. We need international partners for delivery of the treatments beyond our borders. And we also need intellectual support from teams like down at Johns Hopkins or at the CRIC in the UK, who are world-leading experts to amplify our efforts, validate them, and support our findings.
And what's really unique about our network is that it's grown both organically and exponentially, which I think is a really rare combination and a testament to the leadership that we have at the Goodman Cancer Research Center, which starts with our director, Dr. Morag Park.
And that's really what's special about the way we work. There's free flow of information from the clinic to the lab so that we can feed back the information that we're learning that is world-leading data back to our patients, back to our population here in Montreal and Quebec. So I hope this gives you a little bit of perspective on what we are doing to help lung cancer patients live better and survive longer.
Thank you very much for shedding light on this complex area of research. Now it's time for our Q&A, and we'd like to hear from you, our viewers, what you are interested in. So blast away, ask your questions. Welcome, everyone. We sure learned a lot there. It's kind of tempting to say it was fun, but when you talk about lung cancer, fun doesn't seem to be the right word. But it certainly was very, very informative. So now it's time for you guys to get involved and I'm sure there are many, many questions that arise. We'll try to handle as many as possible. And we're going to get the ball rolling here with, I think, a question that probably has crossed many a mind. What is the difference between chemotherapy and immunotherapy? Maybe John can handle that one.
Sure, Dr. Joe. So it's a question I get actually often from patients. The way I explain it is that chemotherapy and immunotherapy are categories of medications that have anti-cancer activity. And essentially, chemotherapy are a long list of types of chemotherapy that are out there. Most are given through the vein, but they can also be delivered by pill form. And what's common amongst them is that they're directed towards rapidly dividing cells, and they're aimed to kill the cancer cell itself. And they have some off-target effects on other cells in our body that are rapidly dividing. Immunotherapy is a newer category of medication, and it's really we're in the first generation of these drugs. And what's special about them is they are able to wake up our immune system so that it can go ahead and kill the cancer cells itself. Because a lot of cancers have a tendency to put our immune system to sleep and prevent it from doing what it should be doing every day, which is chewing up and killing mutated, altered cancer cells. And so those are the two classes. There's also targeted therapies that are aimed at specific changes in molecules that are present on cancer cells, and there's a growing list of these that are highly active and are really changing the cancer landscape. So those are and all of these drugs are aimed at getting cancer wherever it is in your body. So,
very, very good. One of the classic studies in epidemiology was the linking of lung cancer to smoking by Sir Richard Dahl. And I mean, that is something that is taught in all epidemiology courses. But it brings up a question. Obviously, not everyone who gets lung cancer smokes. So how is that? What else can cause lung cancer? Maybe we'll go to Logan for that one.
Sure, it's a great question, Dr. Joe. So we know that about 85% of all lung cancers are attributable to modifiable risk factors, making it really one of the most, if not the most, preventable type of cancer out there. 70 to 75%, depending on where you're looking up your statistics, are directly a result of tobacco smoke. And then there's about another 10 to 15% that can be attributed to things such as air pollution, radon gas, asbestos exposure. More recently, there's some data looking at diet and physical activity. And then, of course, the last 10 to 15% are unknown. Now, having said that, cancer is a genetic disease. And the fact that we know that tobacco causes genetic mutations, which gives lung cancer, may somewhat mask some of the genetic underpinnings of lung cancer, because the majority of the patients are getting genetic alterations from cancer. Now, if none of those people smoked, we may have a better idea of what's happening genetically in the population of people that are getting lung cancer. So I think that sort of gives you a little bit of a landscape of all of the major reasons why people that don't smoke may be getting lung cancer.
Where does smoking cannabis fit into this?
I get that question actually very often from patients in my office. It's a common practice. I think any inhaled smoke, be it from tobacco or other substances, we know in countries where people cook with charcoal or wood, or people who in the workplace are exposed to these kinds of fumes in high quantities are much more at risk of lung cancer. Really, any inhaled smoke is potentially damaging to the lung, not just for the reasons of cancer, but for other lung diseases like COPD, emphysema, etc. So I don't have any moral objections to smoking cannabis, but I can't advocate for my patients to do that. I sort of tell them try oral approaches or things like that.
How often is lung cancer primary as opposed to having metastasized from somewhere else?
So, yeah, sure. Well, to this day, although lung cancer is accounting for such a major societal burden in terms of cancer-related deaths, and that we have proof that screening for lung cancer using CT scans reduces the rate of death due to lung cancer. We don't have, up until just very recently, programs in Canada that allow people to get screened for lung cancer. And what that means is that the majority of people show up, get diagnosed as a result of advanced disease, so stage 4, 3 or 4 disease, presenting, because it's starting to cause them to cough up blood, get very out of breath, lose weight, all the usual symptoms of cancer. And so still to this day, a little more than half of patients will present with disease that's stage 4 or advanced in a way that we don't typically think of being able to cure, but that's changing. And what's been special about the screening programs is that we flipped that in the places where it's been done, the proportion of people presenting with early stage, what's curable by surgery or targeted radiation, has totally flipped on its head, so that far more people who are screened will be diagnosed with early stage cancer.
Is a lung transplant ever done for lung cancer?
Well, I wouldn't say it's a standard care of care, and the simple reason is that it's been done in very peculiar scenarios, not to much great success. And part and parcel of a lung transplant is that you have to suppress the immune system and to tolerate the graft from the donor. And when you do that, cancers run wild. So it's not any kind of transplant isn't a very good thing for someone who's prone to developing cancer or has cancer, because it tends to grow out of control. As we said with immunotherapy, maybe Logan can elaborate a little bit on, the yin and yang of why a transplant patient might have a cancer that would grow out of control, and why we use immunotherapy to treat people with lung cancer.
Maybe Logan, also you can comment on why it is that immunotherapy doesn't work for everyone.
Yeah, so I think it's important to keep in mind that, again, 50%, as John mentioned, of patients that present with stage 4 lung cancer, that means that it's already spread to other organs and tissues, which means that they're not going to be a candidate for surgery once it's spread. So, with respect to the transplant, most of the early stage lung cancers, John is able to remove from the lobe, and so a transplant wouldn't necessarily be the most appropriate fit for that. In terms of immunotherapy, it's a great question, why some patients respond to immunotherapy and some don't. I think, the easiest way to understand it is perhaps to relate it to what's happening right now with COVID. Our immune systems have evolved to be very specialized and very unique. And throughout our lifetimes, depending on where you live, what you're exposed to, you can have very different activity in your immune systems. And we see that with immunizations for COVID. We see that from people that are being exposed to COVID in addition to other viruses. Some people will, who have children in daycare, will be sick very often, and then that will enhance their immune system. So when it comes to immunotherapy, the whole premise behind it is simply to allow your immune system to attack your cancer. But again, everyone's immune system is different, and some patients seem to have a better ability to do that than others. And I'd say that's sort of the generalized view of that.
Yeah, I know that one question I get very often these days is, what can I do to boost my immune system? People want to know. And I think, we have to clarify that the immune system is a very, very complex system with many organs and cells involved. And boosting the immune system is not always welcome. If someone has an autoimmune disease, you don't necessarily want to boost the immune system. So, there's the question that comes up so much these days of, what can I do to boost the immune system against COVID is really a simplistic question that just cannot be answered. Anyway, I have a very interesting question.
If you don't mind, Joe, I mean, I think it is a central question to understanding why immunotherapy doesn't work in some people, in the sense that for immune cells to kill cancer, they need to be able to come into contact with them. And there are all kinds of things that people are looking at to enable that. So by looking at molecules that will allow these immune cells to more easily penetrate into the cancer's microenvironment. And what's fascinating to me is that, some scientists have shown that the kinds of bacteria that live in our gut, and this is particularly true with lung cancer, tend to explain, well, we don't know if it explains, but some people with certain bacteria are much more likely to respond to immunotherapy than others. And we're not totally sure why, but it might be because those bacteria express things on their surface that are similar to what's in the cancer, and that your body may already be kind of ready to tackle those anything that bears those markers. And so people are looking at foods, specific oral supplements that may make immunotherapy more effective. And so this is very exciting.
It's a fascinating area of research. I mean, the microbiome, and there's so many things, I think, that the microbiome influences in our body. Okay. And I don't think we appreciate those bacteria as much as we should. The fact is that we are actually more bacteria than human, right? We have more bacterial cells in our body than the human cells. So we should appreciate that. Anyway, someone wants to know.
One thing, Dr. Joe, can I just jump in here? Because I think it's important to your point about hyperactive immune systems. So everyone wants to have this very active immune system, but you have to also keep in mind that, 2/3 of all patients that get immunotherapy also have immune-related adverse events. Your immune system attacks your normal healthy tissue. And there's definitely a balance that has to go on there in terms of having, the people that get more adverse events, the immunotherapy does better for their cancer, but then they suffer more side effects. So, I just think as a general understanding of immunotherapy, it's great to boost your immune system, like you said, and you want to have this active immune system against your cancer, but that sometimes can come at a cost where, your immune system going haywire can also attack people, and you end up with all sorts of adverse events. What is their cost benefit to having a very active immune system?
Well, of course, we're seeing this with the vaccine, right? Some people have have an immune reaction to the vaccine itself, others don't. And there's all this talk about whether or not, if you have a significant reaction to the vaccine, does it mean that it's going to work better for you because that reaction is a demonstration that your immune system is working? Is there any truth to that?
Oof, I wouldn't venture a guess at this point. I see all those dots behind you, and I wouldn't want to be, in the same category.
All right. Someone wants to know, as a patient, how can I contribute by donating human specimen for your research?
Wow, that is a fantastic question, and I think it is probably the most important thing you can do. Every time I go and give a talk, I always thank at the end of my talks, because I do a lot of personalized medicine research, meaning patient-oriented research on clinical samples. I always thank the patients at the end, because, most people, when they're diagnosed with lung cancer or any cancer for that, right, it's an extremely scary time for them. Most people don't have the background in medicine or science, and yet they're still willing to donate their specimens for research purposes, regardless of knowing what's going to happen to them. So it is the single most important resource that we have as researchers. The overall question of why immunotherapy works for some patients and doesn't work for others, the only way we can figure that out is by getting samples from patients where it did respond and patients that it didn't respond. And that's how we study these in the lab. So donating, blood, or donating, your surgical resection for research purposes, ultimately is what leads to breakthroughs. It leads to, you know, getting better therapies for the next generation of cancer patients. So I think it is the most important thing that you can do as a, you know, one of the most important things you can do as a patient.
How would this happen on a practical basis? Like someone like this who's willing to donate something, how would they approach it? Who would they contact?
So usually patients are approached by various research coordinators in the hospital, and you know, our university-based hospitals are very research intensive, and so almost regardless of the disease you have, it's likely as a patient that you'll be asked to participate in some kind of study. Some studies are treatment related, but a lot are don't have much to do with treatment, but obtaining samples for people with that particular disease, and it may require extra blood draws or tests, but it doesn't always mean that it requires extra things. And so, as a patient, I think it's worthwhile to inform yourself of what's involved. And understand that your privacy will be protected with you know, the most rigorous standards, and that the participation in those kinds of efforts is what changes outcomes for people. Now, someone who, you know, we often need people to donate tissues, bloods who aren't affected by the disease as controls, and so. There are scenarios where we may reach out to people for that sort of thing. But
there's one question that has come up repeatedly here. What are the first symptoms of lung cancer?
Yeah. So the trouble with it is they could be also with a public lecture like this. You know, I don't want everyone going to their GPs worrying about having lung cancer. But that's what's difficult, is that often the symptoms could be like a cold, you know, or a recurring cold. They may have a cough that's worsening. Things that are you know, alarm bells should go off, as if you're coughing up blood, if you're getting more and more out of breath, weight loss that is unwanted and out of control, those sorts of things. But what we're trying to do with screening programs, as much as possible, at least, is to shift things so that people who we've identified as being at risk for this disease have access to ways of detecting the disease early, so that we're not waiting for the symptoms to show up. Because, as I said, sometimes certainly when symptoms have shown up, that's when things are harder to manage. Not impossible, just more difficult.
Is there any advancement happening in gene therapy? And does CRISPR have a potential role in lung cancer?
It's a good question. So I would say not directly in patients as of yet. You know, there's not many applications or you know, of course, given the ethical concerns of CRISPR or gene editing in patients, but definitely in the lab. CRISPR has become a routine tool that we use in the lab, where we can identify from patients what are the major genes that are associated with driving lung cancer. And then we can take cells in the lab and we can modify them using CRISPR to have the exact same genetic alterations that cause cancer. So we can turn cells in the lab to mimic the cells that are in patients, and we use that as a tool to then study how they work in other models, such as, you know, mouse models or marine models for studying. So not directly in terms of gene therapy in patients as of yet, but definitely a hallmark tool used in the lab to edit genes and to study them.
Yeah, I was fortunate to work with the person who invented the first gene therapy for lung cancer, Jack Roth, down at MD Anderson, and the objective was back in the late 80s, early 90s, and the idea, about half of lung cancer patients have a mutation in a gene called P53, and the idea was to try and put in a normal P53 gene in tumors, and it was a great idea, but didn't work out. And so you know, as Logan is indicating, it's extraordinarily complex to bring that kind of therapeutic approach to patients.
Here's a sort of a personal question to both of you. How has COVID affected your research and your practice? What has changed for the better and for the worse?
Oof, we don't have enough time to list all the ways it's changed our lives. Look, I try to be optimistic, and I think it's brought a lot of good into our way of helping people. Certainly, I find by opening up telemedicine and all this technology that existed, we just weren't using it in our hospitals. I think we've helped patients have a more livable trajectory of their disease. They're not coming into the hospital uselessly to do things. If they're coming to the hospital, it's because they're coming to either have a test or something that they need to be in the hospital to have done. So I think it's been positive in that respect. Now, of course, all of the fears, legitimate fears about seeing a doctor, consulting for problems, have been massive. I think there've been a lot of patients who've not shown up to medical attention for fear of exposing themselves. It's created horrible scenarios in the hospital, you know, with quarantine, people not being able to visit their loved ones. So it's been a really challenging year for everyone, healthcare workers, patients alike, scientists. But maybe, Logan, I don't know, I find it's connected us in ways that we've never been so connected around the world. And so, I mean, that's
I couldn't agree more. I think, you know, the initial, from a research perspective, of course, you know, especially in the early days, a year ago, you know, it was challenging because, you know, we try and collect and bank specimens from patients, but we didn't know if they were contaminated with COVID or not. Nobody really knew what was happening. So, you know, it stopped us from doing that for quite a long time till we could figure out how to test all the samples, test all the patients, and get that together. From a connection perspective, though, it really has been a blessing. This online interface that, you know, we curse sometimes, you know, sitting at home and not getting that personal connection has opened up a possibility for us to meet with other labs and groups throughout the world. We have weekly lab meetings with groups in Europe, in the US, and things that you would never consider, you know, bringing other investigators or teams into your lab meeting, because usually they're just in a small room in person at your center. And so it's really expanded our collaborations, our connections, and in that way, I think it's truly been a blessing.
Well, we're just about out of time, but I think we've learned a great deal about lung cancer. And I think we can also say that there is light at the end of the tunnel. And research is starting to pay off, but research, of course, is long, and you have to grind through a lot, and it doesn't happen instantly. But it's really fascinating to hear both of your experiences, and it kind of makes you proud to be associated with McGill, because we see that McGill is at the forefront of research and of medical practice. And of course, the university has a long history of that, and it is being kept up. And I think that our audience has enjoyed your talks and the answers to the question a great deal. And I think given them an insight into how the world of research works and also how the world of medical practice works. So I want to thank both of you for your insight and for all of the fascinating information that you provided. And the Goodman Cancer Research Center, of course, is one of the prime cancer research centers in Canada. The work goes on. They need your support. And certainly there will be more of these presentations where we will find out exactly what the researchers are doing. So guys, thanks again very much. And thanks to the audience for sitting in on this. And I hope that you've been informed and a little bit entertained as well. So until next time, so long.